Okay, so the question was, on the day of the test, do we come to class? On Friday, which is the day of the individual exam, you will come to class, and we'll do like a review session, and we'll finish up membrane transport. And then the exam will open 5 o'clock on Friday and close at 11:59 on Sunday. Guys, I cannot stress this enough. Do not wait until 11 o'clock on Sunday night to start the exam, because if you have proctorio issues, you're not going to be able to get a hold of me or Dr. Arnett or IT at 11 o'clock on a Sunday night. So please, 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 you don't have to take it on Friday. I'm not saying that. I'm just saying don't wait until the very last minute, because as sure as you do, something will happen. Okay? Good question. Is there anything else? How many questions are on the test, and are they multiple choice? Okay. So, on the individual exam, which you'll take over the weekend, it has, I believe, around 30 questions, and I believe she gives you around 50 minutes, so like a normal class period to take it. You probably won't need the full time. And it is all multiple choice. So the whole exam is multiple choice, both the individual and the group. Now, the group exam is on Monday, and you will not come to class on Monday because you'll be taking a group exam over Collaborate. Now, the group exam is pretty much exactly the same test as the individual, but a couple more questions. So I think there's like 29 on the individual, and I think there's like 33, 34 on the group. So it's virtually the same test plus a few extra questions, and you get to take it with groups of 4. Sadly, y'all don't get to pick your groups. That's pre-assigned, but you will get to talk out questions, and maybe you know an answer that somebody else doesn't, and somebody else knows an answer that you don't, and the group exam is factored into your overall exam grade by 15%. So your overall exam grade is 85% the individual, 15% the group. And I know that sounds so scary, and you're like, oh my gosh, but I can tell you, y'all, this is my 3rd semester, and the group has never, ever, 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 literally never dropped someone's grade. It has always buffered it. For some people, it is the difference between failing and passing. For some people, it's the difference between a B and an A. It is a good thing, and once you guys see your next exam grade, I'm sure you'll understand, but I know it's very stressful at first. All right, next question. What does the test material start and end on? So it starts not the first day because that was syllabus day, but the very first day of content with the bio theme, so like the evolution, stuff like that. It starts there, and then we'll end on whatever we end on Friday. So it goes from the start of class to Friday. Okay, any more questions? Somebody at least say no, so I know that you can hear me, because I'm paranoid now.
No.
No. Thank you. Thank you so much. I am paranoid that I'm talking to a wall now. Okay, so I'm hypothetically sharing my screen. Can you guys see this PowerPoint?
Yes.
Thank you, guys. Thank you for answering. Okay, so Test 1 Survival Guide. We're going to start with the very first thing we talked about in class, which is level of organization. And hint, hint, I didn't just bold things for my health. So, like, do what you want. So, level of organization, this is a list of biggest to smallest. So, the biosphere is the whole Earth. An ecosystem is all living and non-living things. in a particular area. So say we're talking about the University of Charleston. The ecosystem would be the students, the squirrels, the rocks, the grass, the buildings, the trees, the soil. That would be everything. Now, a community is just 2 or more species in a certain area. So if we're still talking about UC, that would be the students and the squirrels. That would be a community. Or the students and the grass. Just 2 or more living species. Okay? Now, a population is just 1 species in a certain area. So that would be like the human population of UC, or like the grass population of UC. A population is 1 specific species. It's only 1 species. An organism, an organ tissue, that is pretty self-explanatory. An organism is 1 single person. An organ would be like your heart. Tissue is what makes up an organ. So like your soft tissue or your skin tissue, stuff like that. And the cell is the smallest unit of life. So anything below a cell is not alive, like an atom. But anything above a cell, like an organism, is alive. Does that make sense? Okay. Themes of biology. So here we have genetics, which the main thing that you need to know about genetics is just that the chromosomes are the main units of inheritance. Now I'm going to skip down to cell theory. You obviously need to know a little bit more about cells because of the 3 main themes here. Cell theory is the one that we covered for this exam. But the basic thing of cell theory is that a cell is the lowest level of life, which I just talked about. Now, for evolution, do you guys remember, I think it was like the 2nd day of class, she done a moth example about evolution. And it was basically that there was a birch tree, well, many birch trees, not just 1, and they were white. So white moths camouflaged more, and gray moths stood out more, so the birds ate the gray moths. But then the steampunk era happened, and the birch trees turned gray. And then what happened was the white moths were no longer camouflaged, so they were being eaten more. And the gray moths, the population started to grow because they weren't being eaten. Does that make sense? Do you guys remember that? Okay, so the key to this, the main key. Yes, thank you. Okay. So the key to this, the main key, is that dead things don't make babies. And I know that's like, duh, but that is the foundation of evolution. If you're eaten by a predator, then you're not going to be able to reproduce. So when you are looking at a word problem or looking at an example, think, okay, this is very simple. Dead things don't make babies. So which color moth is alive and which is not. Okay? All right, parts of an experiment. I know in lab you guys have beat these to death, and especially for your research lesson 2, I was trying to think, the graph experiment. So an independent variable is a variable that's being manipulated. It's something that I can change. So like time, temperature, maybe it's like days. So that's something that I can change. The dependent variable is what is being measured. So the dependent variable is your y-axis, while the independent variable is your x-axis. Okay, so the control group, that's just a group that you keep the same. So I know in class you used the example of plants and fertilizer. So if I just have one plant that I add fertilizer to, how do I know if it makes it grow more, less, or the same than a plant without fertilizer? You have to have something to compare it to so you can see if you're actually making a change. Okay, now a controlled experiment just says that all variables except for the IV are the same. So if I am talking about plant growth, I can't test a corn plant with a bean plant and then test height, because that's not how it works. A corn plant is obviously going to grow much, much higher than a bean plant, I think. I don't know. I'm assuming. And you have to make sure that they each get the same amount of water and they each have the same soil minus the fertilizer. Stuff like that. Okay. Inductive and deductive reasoning. So inductive is many observations used to make a generalization or an inference. So that is the upside down funnel, right? As is the V. Now, a deductive, remember I mentioned this in my last study session, think deductive, detective. So think, okay, if all of these claims can be taken and come to this conclusion by a detective, could a detective logically figure this out? Then it's deductive. But if a detective would not be able to figure that out, then it's inductive. Now, I would know an example of each, or at least know how to tell them apart. Okay? All right, so next we have hypothesis, theory, law. And I would highly recommend word associating. Hypothesis is testable and falsifiable. Falsifiable might be a better word. It's a prediction or an educated guess. A theory is an attempt to broadly. I would word associate theory and broadly. So a theory is an attempt to broadly explain laws and observations. Okay? So a law is a generalized statement based on repeated observation. Excuse me. And test to predict future events. So I would word associate. Hypothesis, testable. Theory, broad. Law, generalized. Okay? All right, so that is all on Unit 1. Any questions about the parts of the experiment, themes of biology, stuff like that? Okay. Now we are going to move on. Notification. Okay. Yeah, so I will send out the PowerPoint. It might be a little bit later tonight than directly after, but I will 100% always send it out the 9th. It may just not be directly after, like it has to be. Okay, so moving on to functional groups. You guys need to know the functional groups. There is no getting around it. Okay, I don't know what it is. What is it? The audio is weird. Maybe it's my fan? Like a cracking noise. My audio is awful. Cool. Is this better? No. Okay. have no idea what to do. Oh, somebody said yes. Maybe. Okay. Whew, I love technology so much. Okay. I'm going to give this a minute to hopefully reset. Refreshing. Okay, cool. Let me refresh my page. So guys, it might kick me out for 1 second, but I will be right back, okay? Let me see if I can do anything. Okay, guys. Please tell me it's better. Oh, hallelujah. Okay, so if this happens again, just yell at me and tell me to refresh my page, okay? I will not get offended if you're like, hey, your audio sucks, refresh your page, okay? But you will have to tell me because obviously I can't hear. Okay. Now we're going to go back. Can you guys see my PowerPoint? Okay. Assuming that's a yes. Yes. Okay, so functional groups. I was saying you guys have to memorize your functional groups. I'm at a 400 level biochem class, and we're still using these functional groups. Not only is it on this exam, but you will use it in so many more advanced classes you need to know. So, a carboxyl. Well, sorry, let's start with a hydroxyl. So a hydroxyl is just this OH group. So hydro, hydrogen, oxyl, oxygen. Okay? Okay, now let's go over to a carboxyl. A carboxyl has a hydroxyl on it. So this OH right here, that's a hydroxyl. So that's how you can distinguish the two. So a carbonyl just has a C with a double bonded oxygen. So you can think carboxyl has a hydroxyl, or if you think that's like too confusing, which I totally understand, just think that the one that sounds like oxyl, so oxyl sounds like oxygen, the one that sounds like oxygen has the extra oxygen. So this only has one oxygen. The carbonyl only has one oxygen. Oxyl oxygen has the extra oxygen. Okay, now an amino group is just a nitrogen with two hydrogens. Okay, does that make sense? Okay, so making and breaking molecules. So hydrolysis is breaking a polymer to a monomer by adding water. So hydro means water, lysis means to break. So hydrolysis literally means breaking something by adding water. Okay, now dehydration synthesis means that you're making a polymer from two monomers by taking away water. So dehydration synthesis means that you're adding two things together by taking away water. So 2 very important things to note is making occurs at hydroxyl groups. So making is dehydration synthesis. That will always occur at hydroxyl groups, 1 from each molecule. Okay, now breaking occurs at the shared oxygen. And it will be very clear what a shared oxygen is because there's 2 independent molecules with just an oxygen in the middle, and that's usually the only thing that is connecting them. Okay? All right. Now on to our first biomolecule, carbohydrates. So the function of carbohydrates is that they serve as building blocks and fuel. You can identify, I don't know why this is small. Hold on, I'm going to make it bigger. Okay. All right, so you can identify it by the crap ton of hydroxyl groups, or you can simply say it has a lot of oxygen on it. So oxygen, 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 blah, blah, blah. Okay. It also has, what kind of group is this up here? This right here that I'm circling. Somebody tell me what this group is. It's a functional group.
Is it a carbonyl?
Yes, it is. Perfect. Okay. So a carbohydrate is polar because of all the oxygens. So if something is polar, what is it? Hydrophobic or hydrophilic?
Hydrophilic.
Awesome job. Okay, so now this is just some vocab that you need to know about it. A monosaccharide is a monomer. A disaccharide is just 2 monomers linked together by dehydration synthesis. A polysaccharide is just more than 2. So we just get lazy. So after 2, we just stop counting. Now, an example of a carbohydrate is starch. Okay. Now, lipids. All right, so you can identify a lipid by a carboxyl group. So here you have the carboxyl, you have the hydroxyl, or you have the 2 oxygens, okay? And very few oxygens. So you'll see on this chain, you only have 2 oxygens associated with the carboxyl group. Now, you could have more, but you will at least have 2 in every single lipid because of the carboxyl group. So lipids are nonpolar, which means that they're hydrophobic and they do not mix with water. Now, the main structural thing about lipids is that they have a very long hydrocarbon chain, and this chain makes them hydrophobic. So hydrocarbon is technically a functional group, but it's just a carbon with hydrogens on it. That's a hydrocarbon. And it is hydrophobic because the hydrocarbon has no oxygens on it whatsoever. Now, the main types of lipids are phospholipids, steroids, and fats or fatty acids. Now, on to her favorite biomolecule, the phospholipid. Phospholipids make up the cell membrane. They have a polar head and a nonpolar tail. Now, a phospholipid contains glycerol, a phosphate group, and 2 fatty acids. So glycerol, we really aren't that worried about. It's kind of what links the head to the tail. So the head is the phosphate group. And it is what makes it polar because the phosphate group has a lot of oxygen in it. Now, fatty acid, you have one saturated, one unsaturated, and those are the tails. So those are hydrophobic and nonpolar. Okay? All right, now we're going to move on to proteins. So proteins are the most versatile biomolecule. They contain an amino group, a carboxyl group, and an R group. You guys, you cannot determine the polarity of an amino acid without the R group. You can say which one's more polar than the other group-wise because an amino group doesn't have any oxygens, but a carboxyl group has 2 oxygens. So a carboxyl would be more polar than an amino group, but you cannot determine the overall polarity of a protein without knowing that R group, which she won't really make y'all do. Shh. Okay, so the primary level of a protein is also called a polypeptide chain. So if she asks something about a polypeptide chain, she's really just asking about the primary level. And also a monomer of a polypeptide is an amino acid. So all polypeptide chains, so the primary level is just amino acids linked together. So the most basic level is an amino acid. Okay? All right. Levels of organization of a protein. You guys need to know which bonds are associated with each level. Okay? So primary is associated with peptide. Think P and P, primary peptide. Secondary, this is the only one that I cannot find a mnemonic for. Secondary is held by hydrogen bonds. Okay? Tertiary is held by side chain interactions, and it is of course everybody's favorite mnemonic. Terry is a side chick. Tertiary, side chain interactions. All right, now quaternary are multiple polypeptide chains, and they are held by hydrogen and or ionic bonds. So my mnemonic for this is if I had a quarter for every high I heard. So quarter, quaternary, high, H-I. I was stretching, but that's all I could come up with. You do need to know which bonds are associated with each level for every single level. Okay, so now moving on to nucleic acids. So every nucleotide, which is a monomer of a nucleic acid, is the most basic unit, contains a phosphate group, a 5-carbon sugar, and a nitrogenous base. Okay, so you need to know what makes up a nucleotide. Okay, so types of nucleic acids. You have DNA, which is double-stranded, stays in the nucleus, and deals in long-term genetic storage. And you also have RNA, which is single-stranded. It leaves the nucleus, and it aids in gene expression and regulation. So how I think of this is that you have DNA, which is like the big boss. He's the rich guy that sits in his mansion and tells his workers what to do, but he really doesn't do anything himself. He just holds all the information. And he has a lot of money and he doesn't really do much, so he's maybe a little bigger because he's double-stranded. Okay? Now, the RNA, this is like the working man. He's a little thinner because he works all day. Maybe he's not maybe he doesn't have that much money, so he's single-stranded. He leaves the mansion after he gets his orders. He does what he has to do. He works. Okay, so he's the one that actually does stuff. Does that make sense? Okay. All right, so that is all on biomolecules. Does that make sense to you guys? Do you have any questions about biomolecules or anything that we have covered thus far? Let me see if I can get my chat up. Okay. No questions? My audio still good?
You're still good.
Thank you. Y'all, please yell at me if it goes bad again, okay? I will not be offended. If y'all can't hear me, then I'm talking to my wall or my roommate, and they both don't need to hear it. Okay? All right, so now on to the cell. So a plasma membrane is made out of phospholipids. And a plasma membrane is found on all cells. All cells have plasma membranes. Okay, now they're made of, as you can see over here, a phospholipid bilayer where the heads are out on each side of the cell. And the little tails are tucked in the membrane. So the tails aren't exposed to the outside or the inside of the cell. They're just kind of chilling on the inside of that thin little membrane. Okay, so that makes the outside of the membrane polar and the inside of the membrane non-polar. And this is kind of self-explanatory because if you put water on your skin, is it just going to sit there or is it going to kind of soak in? Or if I put water on my skin, is it just going to like repel off and like shoot little water droplets everywhere? It's not going to do that, right? So it's going to sit there and slowly sink in if I leave it there long enough. And that has to do with this semi-permeable membrane, but also that the outside of every cell is polar. Does this make sense? Okay. Now moving on to organelles. So the question. Okay. All right. So now we have the nucleus. It contains cells. It contains the cell's genes, and it controls growth and reproduction through DNA. So the main thing that you need to know about the nucleus is that it houses the DNA. Okay, now the nuclear envelope is the special protective membrane for the nucleus. And this is pretty self-explanatory because if I wanted to mail a letter in the mail, I wouldn't just take my letter with very sensitive information and just stick a stamp on it and put it in the mail. I would put it in an envelope for extra protection. It's an encasing. Okay? So now we have ribosomes, which carry out protein synthesis. We talked about that today, so you should feel pretty good on this, or if not, please reach out for help, guys. That's why we're here. So, but we did talk about most of this today, so if it's still kind of new, that's totally okay. So a ribosome carries out protein synthesis, and it can either be bound or free. So if it's bound to the rough ER, which rough ER, the main thing you have to worry about the rough ER is that it has bound ribosomes to it, so it looks like bumpy. Okay, and then if ribosomes are free, then they're in the cytoplasm or the cytosol. They both mean the same thing. All right. So next we have the smooth ER. They lack ribosomes, but they synthesize all of our other biomolecules. So they synthesize carbs and lipids. Okay, so lysosomes, they are a membrane sac of hydrolytic enzymes. So they basically just break down things. They eat stuff. Think of them like the Cookie Monster or Pac-Man. Think of them like a Pac-Man. They just run around the cell and they eat stuff that needs to be eaten. They break it down. Okay? So now the Golgi, that is like the FedEx of the cell. It will take something in through the cis phase. It will modify it, sort it, package it all up real nice, and put it into transport vesicles and ship it out the trans phase. okay? And the whole Golgi is made up of cisternae, which are just flattened membranous sacs. So I know in class you use the example of like a plastic bag full of like pudding and how you just stack them. That's pretty much the same. That's pretty accurate of what it looks like. It's very kind of flimsy membranes just stacked together. Okay? So now you will need to know, or let me rephrase that. I would know how to identify the major parts just from a basic picture. Just from a basic picture of an animal cell. Know like, hey, that's the nucleus. Here's the mitochondria. Here's like the cell membrane. Just basic stuff like that. Okay? All right. The pathway for protein synthesis. You go from the nucleus to the ribosome to the cytosome or the rough ER. Cytosome if it is free, rough ER if it is bound, then to the Golgi, and then to the transport vesicle and out of the cell. Does that make sense to everyone? Okay. Now this is on to membrane transport, which we have not gotten to yet. We will get to on Wednesday, maybe Friday, but I'm just going to go ahead and just read off my slide. If you guys have no idea what I'm talking about, that's okay. Like I said, we're not even going to talk about it in class until Wednesday or Friday. So it's okay if you don't understand, but I'm just going to read it and give you guys like short mnemonics. Okay? So diffusion just means that you're going down concentration gradient. So diffusion is down. The D's match up. And this is only for small molecules, such as like hydrogen, oxygen, maybe some nitrogen, just single elements. Okay? So now we have facilitated diffusion, which is also down the gradient, but it uses a protein to move larger molecules such as glucose. Osmosis is just the diffusion of water. So it just means that water is moving across the membrane. All right. So even at equilibrium, molecules still move. If a cell is not moving, then it's dead. Okay? All right. So tonicity, you have isotonic, which is just the same concentration on the inside and the outside of the cell. So in an animal cell, isotonic is what you want. So I think of this like iso happy. I know. Guys, I know how dumb my mnemonics sound, but I promise you they work. So isotonic, iso happy. Hypertonic means that the concentration is higher outside of the cell, which basically just means that the cell is going to shrivel up. Hypotonic, I think hypo, hippo. So the higher concentration is inside of the cell, and the cell will eventually burst. So I think the cell gets really big like a hippo. Okay, so when you're approaching a problem like this, don't look at the specific concentrations, because if she puts a number in there, it might like freak you out. So don't even look at the specific numbers. Just look at which one's higher. Just think, okay, if I have 200 and 300, I would just say, okay, the 300's higher. That's the only thing I need out of specific numbers, is which one is higher. Okay, so active transport, I don't really know if we are going to get to all of this because we're just a teeny tiny bit behind schedule. So all I'll really mention about active transport is that it requires ATP, which is the cell currency. So ATP stands for adenosine triphosphate, which is just the currency of the cell. It's how it runs. It's how it like, it is. When we talk about cell energy, we usually are talking about ATP. And ATP is used for energy when you break it down through hydrolysis. And with active transport, you are moving against the concentration gradient. So you're going from a low concentration to a high concentration. That's all I'm really going to talk about with active, because I'm not really sure how far she's going to get. Okay? All right, so that's all I have. So let me stop sharing my screen. Possibly. Okay. What questions do you guys have? Okay. Does anyone have any questions? Is there anything that's just not really clicking? Yes, so I will send out the PowerPoint. I actually have plans at 6. So I probably won't send it out directly after, but I promise you will get it tonight, okay? I promise. All right, no problem. Yes, so Fallon is going to have a study session. I'm not sure what day, but she will have a study session this week. All right, thank you guys so much for coming. I just took a picture of your names for attendance. So if you don't have any other questions, feel free to log off. But if you do, guys, please stay and ask them. That's the only way that you're going to get help, okay? All right, you guys have a great day.